Okay, so this repressor protein is a dumbbell-shaped protein, 132 amino acid long. Okay, and you have there are two termini, N terminus and C terminus domain. N terminus is from 1 to 92 amino acids, and this is the DNA binding domain, whereas C terminus is from 112 to 132 amino acids, dimer binding domain, and this central rod portion is there. So this repressor, the binding site for the repressor is the operator region. So this repressor will come and bind the first few molecules of repressor which have been synthesized by the RNA polymerase transcribing CI. RNA polymerase which is bound to promoter PRE. First few molecules, once they are synthesized, they will go and occupy this operator. So what does it mean? Once the operator is occupied, they are making sure that RNA polymerase doesn't bind to this promoter. RNA polymerase binding to this promoter doesn't take place. RNA polymerase binding to this promoter doesn't take place. Okay. But they're making sure that you don't have transcription of CRO. CRO cannot be transcribed because CRO is what? CRO is an anti-repressor. CRO is an anti-repressor. Please understand, see the strategy. First few molecules of repressor, they will पहले जाके वो क्या करेंगे? They will go and occupy the operator on the right hand side. Okay, and as a result of that, because of the overlapping nature of the promoter and the repressor, RNA polymerase, bacterial RNA polymerase cannot bind to promoter R, and therefore the transcription of CRO will not take place. Please remember, CRO codes for an anti-repressor protein. So bacteriophage has made sure that CRO is not expressed. This is what is called as establishment. Once it has made sure that CRO is not expressing, then repressor synthesis is done through what is called as the maintenance promoter. There is a promoter called PM, maintenance promoter. Okay. So maintenance promoter, RNA polymerase will bind to this maintenance promoter. Okay, its orientation is also like this, and it will transcribe CI. It will transcribe CI, and the CI will be translated, and you will have this repressor protein again synthesized. So now these molecules, after the first few molecules of repressor, all other repressor molecules, whatever are synthesized, they are synthesized by the transcription of the CI by RNA polymerase starting from promoter PM. Okay, first few molecules are synthesized by the transcription of CI by RNA polymerase starting from PRE. Remaining, once it has been established, lysogeny has been established, once first few molecules have occupied this operator, making sure that RNA poly doesn't bind to PR and there is no transcription of CRO, further synthesis, further transcription of CI will take place from promoter PM. RNA polymerase will bind over here and will transcribe CI, and you will have the repressor protein. Okay, and this repressor protein will be then will be maintaining the lysogeny. Okay, now this repressor protein, its level has to be precisely maintained. Why precisely maintained? Because it is to be maintained very precisely at a particular level, because if the repressor protein is less than the threshold level, please understand, if the repressor protein is less than the threshold level, then in that case, there is always a possibility of premature shift, premature switch to lytic pathway. Please understand, lysogeny is maintained only by the expression of CI or CL. It is the only gene which is expressed during lysogeny. And the gene product, the repressor protein, 26 kilodalton dimer, dumbbell-shaped protein, it is responsible for maintaining the lysogeny. It is responsible for the bacterial cell, the bacteriophage lambda to remain in the lysogenic phase, lysogenic pathway. Okay. So if the amount, the level of repressor protein is less than the optimum level, then there is always a possibility that even when the bacteriophage doesn't want, it can prematurely switch to lytic pathway because there is not enough of repressor protein. Okay. But if the repressor protein level is more than the optimum level, so this is the threshold. If it is less than that, then there is a problem. And if it is more than that optimum level, then there is also a problem that bacteriophage will find it difficult to switch to lysogeny even when the host has become inhospitable. What we have been saying, when the host becomes inhospitable, bacteriophage as an option, it can switch over from lysogeny to lytic, make progeny, kill the bacteria and liberate the progeny. But when repressor level is more than the optimum level, there's too much of repressor protein, it will not allow that switch from lysogeny to lytic. What does it mean? It means that this bacteriophage DNA, means this particular bacteriophage genome, will also die along with the host. Host is dying, host has become inhospitable. Okay, host is going to be destroyed. So along with that host, this bacteriophage DNA will also get lost. So these are the two possibilities. Please understand, and therefore for this reason, this repressor protein level has to be maintained at a very optimum level, at a very critical level. All the time it is maintained at that particular level. If it goes below that, there is a possibility that there is a premature switch. And if it goes above that, then bacteriophage will find it difficult to switch to lysogeny, even when the host is no longer a safe haven, even when the host is in danger, it will not be able to switch over to the lytic pathway. So how do you regulate the level of repressor protein? So what nature has done, nature has devised a very simple but elegant design that it has put the level of repressor under its own control. The level of repressor protein is under its own control. What we call it as autogenous control. The level of repressor is under its own control. We call it as an autogenous control. Okay. So we have to understand this. Now I will be expanding this particular region further. How this repressor is under its own control. How it regulates its own level. How repressor protein regulates its own level. So what was found out is that, you have this CL in the middle, and then you have a PM, promoter PM. Okay. And then what was found out is that there are 3 repressor binding sites on either side of the CL. These operator regions on the right hand side and on the left hand side. Each one of them in fact have 3 repressor binding sites. So I'm making those 3 repressor binding sites. So this is site number 1. This is site number 2. This is site number 3. Similarly, on this side also we have 3. This is 1. This is 2. And this is 3. Okay, and they are referred to as OR1, OR2, and OR3. Similarly, this is OL1, OL2, OL3. Operator on the left hand side, site 1, site 2, site 3. Each site, repressor binding site, is around 17 base pair long.